basically, so I just want to give like a little bit of introduction about my talk, and then I'll discuss about like what I have done, my postdoctoral works, and a glimpse of what I'm doing here. So like we all know about this drug discovery phases, different phases, and so it starts from that hit validation, like kind of from target validation to ultimately end up on phase 4 clinical trials and then FDA approval. So it takes a lot of time, more than 10 years, and like more than $1 billion, $1.5 billion to take one molecules developed to the markets, and like and this cost and time is hugely like on higher end. It's mainly because of the failures. 90% of this like drug discovery programs gets failure in industry or like even academia. So that's like now for industry, like one of the major aim is to like how to minimize the time and like how to like reduce the cost. So now to do these things, there are the two steps. So presently there's efforts are ongoing to optimize. One way is that kind of lead hit to lead optimization and further like preclinical study, that stage during like your whether it's a biological molecule or like it's a small molecule, using a lot of computational like algorithm or like computational nowadays like machine learning methods to like generate using basically reinforced learning and like random forest learning to predict new molecules which will have better properties, so that we do not have to test a lot of thing in like in vitro like in our test lab. And another thing is that if you get a good starting point from like screening technology, and then conventional screening technology, this basically it's a high throughput screening technology which has its own limitation and like its cost is still bit high. So there it comes a new technology. It's called like DNA encoded chemical library technology. So today basically I'll talk about this one, how we can use this technology in our drug discovery program. And each technology has its pros and cons. So basically I will discuss all these and go through. And then for like take home message of this talk will be like now it's on you, like whether how it is going to decide like whether this technology can be applied in your programs, in your research and all this stuffs. Okay, so now before going into let's okay, yeah, so can you see my first slides?
Yes, yes.
Yeah, okay. So before like going into my talk, so first of all I'd like to discuss about the drug targets. So basically, our aim was to like discover library against NAD binding proteins, basically which are biologically important proteins. They are involved in several biological pathways, and basically they're involved in agings and also like longevity, that pathway, and also DNA damage pathway. And these NAD binding proteins, and these class of proteins generally they call PARPs, poly-ADP ribose like protein. They accept these PARPs basically transfer poly-ADP ribose to acceptor protein, and SART twins it removes post-translational modification. So now if you look at this like probe discovery, so like that majority of the protein that has been studied at SART twin 2 and for this like PARP1 along this family. So basically in this plot, the med score, that is the number of studies have been reported, and this is PIC, this is the IC50 or like log IC50 of these molecules, and the sphere size it represents that number of compounds reported in databases. So like basically PARP1 and SART2 like number of compounds that have been reported more as a, so it's basically it says that there is a biasness in probe discovery or like inhibitor discovery. Why? Because this comes from the like associated cost with the like existing technology such as high throughput technology, kind of this high throughput screen. So generally for high throughput screen it's a discrete screen, like you need a expensive robotic facilities and also like expensive skilled members, and here like millions of compounds they are screened individually in their individual biological assay like in plates. So like it cost, if you want to like screen a million of compounds, it will cost around like 2 billion, 0.4 to 2 billion depends on. your like library type, and the number of compounds you can maximum go, it's like 2 million. And another limitation is you need a functional assay, like that protein should be kind of functional, it should give a readout. And time, space, all these things are there. So now here comes this Dell technology. So basically this is the high throughput, this is all compounds are in your plate, and you can see this picture, this is the like high throughput compound like facility component management at Bayer. You can see you need a huge like building to store when you have like 10 million compounds or to 5 million compounds to store that. But corresponding to that, here this is the Dale you can store here in like in tubes. So it's like 1.5 ml tubes. It will be in solution. It could have like 100 millions compounds. So that I have worked like more than a like 130 million compounds. Still like it is in tubes that will be in the solution. And how? So what is that library? So basically, it's a library of small molecule, and each small molecule attached to a DNA sequences, and this DNA sequence is readable. So basically, kind of barcode. So you are going to supermarket, you are buying something and scanning it. Okay, I'm getting the bill. That kind of thing, because it's a kind of pool here, and yes, barcode is attached. So now for this library of small molecules. So one thing is that this number you can go to like millions, like billions, even trillions numbers are also reported. Now we look at this general structure of this Dale. So this is the general structure. Here you can see this DNA, and this DNA is attached to a small molecule. And this DNA has different regions. So when we are designing the library, so basically we'll construct these things like primer, code one, and this code, and then again primer region. And this small molecule, it is attached to a linker, and you are synthesizing this small molecule based on your chemistry, different chemistry, and it has a like this pool. Now this is the like entire technology. So basically you starts with a kind of small molecule library like this pool that is in a tubes, and then you pan that library. So basically you have a target protein which is immobilized, and then you are incubating that library against that target. Now these non-binder, which is non-binder, they will be washed away. So now these whatever the binders, they will be eluted. So now you have this library, then you do PCR amplification to amplify this DNA, and then you do like deep sequencing, next generation high throughput sequencing, and then you analyze this sequencing data. So basically which will reveal that which structure has been enriched and that has the most affinity against your target protein. So what are the like cons here, like pro against pros? So it has like rapid, inexpensive. So basically you can target like lots of protein, I would say like 10 to 12 proteins in 2 to 3 hours, and then the sequencing that and this downstream PCR amplification that takes around like 2 weeks. So like by 3 weeks to like 1 month time you can get your screening result, where like a screening campaign, corporate screening campaign, it can take more than a year. And another thing, like, you need very less amount of protein, like micrograms of target protein, and there is like no requirement of functional biochemical assay. Okay, so now we'll look into the one example. So this is a one library, like two building block library. So here two fragments are attached with a linker like X066 and Y469, and this is the like screening fingerprint of that library. So this is the like x-axis, and this is well, like enrichment score, and this like y-axis. So now if we look at this library, so like each straight line, it is like one molecule, and their height is maximum, means their enrichment score is maximum, so like their affinity kit is like lower. So the only Y469 fragments containing molecules that have been like enriched here. So others molecules have not been enriched. Now, if we consider that, okay, when we are designing this library, so this fragment is not present. So that means this entire screening campaign will be failure. So now to like how to overcome these things. So to make much more successful screening campaign, so there are two approach. Basically, you can do synthesize a large kind of ultra large DNA encoded chemical library which was the focus of this like field earlier, maybe 10 years ago, but now the focus has been changed because there is another parcel like bag and baggage comes with ultra large DL, because when you synthesize ultra large DL, so like basically you generate a lot of synthetic inhomogeneity, so that makes your downstream like heat validation like very tedious, and sometimes it translates into very false positive rates. So that's a one of the another approach that we proposed first, like that could be a like synthesizing a target class focused library. And another things for this approach that driven us, like when we are like academic, so we wanted to apply this technology in academia, because to still compared to high throughput screening, this like technology is cheap, but still it is on higher side when you compare to resources in academics. So that's why like our thought was to can we synthesize a like small library, so that will be like focused on a chemical space, and still it can deliver the same output when we are screening a like larger library. So and we like pioneer this term kind of, it is the target class focused DNA encoded library. So basically this was our like thoughts. So if you look at these Dale structures, so this is the chemical space, and this is the properties of drug targets, and this is the like chemical space for target ligand. So basically our thought was we don't need to synthesize the structures around all these spaces, because if we can synthesize only these structures here and with low cost, and we can like deliver the same output. That's, so basically if you look at this chemical paste, like this rectangular space, if you consider the entire chemical space, it's like very vast, and this is like entire space coverage by diverse biological targets. So theoretically you need 10 to the power 63 molecules in theory. So basically it's kind of, you can say it's one fit all approach. So basically if you want to synthesize one library that has 10 to the power 63 molecules, but basically practically it is impossible. So you don't have this that numbers of electrons in earth. So our approach was, if this is the target class protein, we'll build our library around this like target class, and like that will be cost effective, and we'll see like how our screening campaign looks like. Okay, so like with these things in mind, we went ahead to design our library. And as I already discussed about that, our target protein is like NAD binding proteins. Okay, yeah. So is so is and does anyone have any questions till now?
No, Dr. Dana, you can proceed. If
Okay,
there have any question, we might ask at the end of the session.
okay. So this was our strategy for targeting like NAD binding pockets. So if you look at the structures of our like previous result, because Dell 100K, so we got a hit against tankerase 1. So tankerase 1 basically it's a NAD binding protein. So and that library which has like 100,000 molecules, so with 100,000 molecules we only got like one hit which has these carboxamide structures. So that was one striking features. Then we looked at the like key interaction of NAD plus and like this or PARP like NAD binding proteins. So so if you look, this is the structure of the NAD, and this has the nicotinamide, this carboxamide bond, and this catalytic side, this glycine 863. These are kind of donor acceptor interaction. So it clearly says this carboxamide that's like very important for this to develop molecules against this class of proteins. And another thing, if you look at these existing PARP ligands, all these ligands they have like this either exocyclic or cyclic this carboxamide. So based on these things, we decided that okay, we'll enrich our library with structure like they have carboxamide functionality. So based on that, now like we have to think about the chemistry, like what chemistry will are going to do. So now if you look at these things on DNA chemistry, so like different kind of chemistry have been reported, like all kind of in solution chemistry, not has all been reported, but majority of things. But still that major portion is amide formation. So why amide formation? So because it's a robust reaction with good yield, and you can purchase lot of commercial building blocks, like kind of diverse building block which are acids. So basically you can like increase your chemical space. And another thing is that it's a mild reaction condition, so your DNA damage will not happen. So basically when you like get the result by DNA sequencing, so during doing chemical reaction, like if your DNA is damaged, so then you are not getting the like exact result. So that's why integrity of DNA is very important. So and another thing is this, all these reactions should be water tolerable. So like that's why amide bond formation is like very robust reaction in terms of del synthesis still now. Another thing now that what kind of like conjugation we are going to follow. So basically if you look at this synthesis methods, two methods are available. One is like liquid phase synthesis and another is solid support synthesis, and we decided we'll go through this solid support synthesis. So one thing is that with solid support synthesis you can use lot of organic solvents. So basically you can use wash all your reagents. And during like solid phase synthesis, basically your nucleobases on DNA, they are protected on CpG beads, so there are very less chance that your DNA will get damaged. And basically you can use excess reagents, and that will wash away, and purification will be easy. So now we tested the scope of the reaction, like how our reaction amide formation is behaving. So basically we started with the dummy DNA, is a hexa T DNA and protecting group on, and this is that amine functionality here, and this is the beads. So we like simple HDU IPA coupling. So you have this molecule and deprotect with AMA deprotection. So basically when you AMA, so this bead will fall off and this protecting group will fall, you will have this entire this construct. Now there is question that basically with that we said that we'll enrich our library with carboxamide. There is a high chance that we condition with AMA. Basically, it's a ammonium hydroxide solution. So there, this could be cleaved. So now we have to check like what kind of like building blocks we can use in our library. So to do that, so basically we purchase lot of building blocks, and before purchasing, we did chemoinformatic analysis so that it could be in our drug space. So we tested around like 500 or 600 building blocks. So and categorized them according to their like percent of yield, how many percentage like their conversion rate. So you can look at this like this kind of molecules like which have like cyclic amides, their mass are correct and their behavior was very good. So we enriched our like max library with maximum numbers, kind of this kind of structure which are like cyclic amides. And suppose this kind of structure, suppose with exocyclic, we see some cleavages are there. So basically we avoid those building blocks, and there are others like this kind of we did not see like any correct mass. So basically this kind of analysis before synthesizing your library you have to do with all your building blocks, so that you can know that how they are going to behave when you are synthesizing library. Because when you are synthesizing library, like on second step onwards, you do not have proper like purification, because it's a pool. Okay, so now this is the sum of the HPLC stresses, and I don't need to discuss about details here. And then I'll talk about the design. So this is our design concept. So left hand side, this is the structure of like nicotinamide. This is, you can see this adenine in blue and this nicotinamide, this is in red, and they are like bisphosphate, there's linker. So to mimic that NAD, what we did, we mimic the entire this hydrogen bonding pattern, donor acceptor interaction, in enriching this like exocyclic kind of or cyclic amide building blocks here. That is one fragment. And there's and second position we have attached like another set of fragments which are basically generic kind of drug like fragments, and some of them are also known like kind of adenine binding pocket, adenine binder. And this structure, you can say this 2,3 like cyclopropanamide, this is not the exactly mimicking this structure, but we use this kind of, it will give you like flex, like not hugely rigid or not hugely flexible. It's a kind of dual display. So the based on the cyclopropanamide, and this is attached to a DNA. And this first cycle we have like around 158 pharmacophores, and the second cycle we have attached around 369 pharmacophores. So in our total we have like 58,302 encoded compounds in this library, and we are calling this library is NADL because it's a NAD binding pocket targeting library. So now if you look at this, so this DL synthesis, so basically it's a like combinatorial split and pool synthesis. So if you go, as you increase the number of cycles, and you can see the library members, they'll grow like exponentially. And there are several methods for synthesizing this DNA library. So most popular methods that also like industry also use, it's a kind of DNA record, it's called DNA recorded. Like Professor Richard Lerner and this Professor Dario Neri Barrymore from GSK, they like pioneered these methods. And there's another method, it's called DNA templated. It's from the David Liu Harvard. So this New Evolution is his company, is acquired by Amgen. So this is basically used for peptide-based or peptidomimetics library, and this library has a lot of application on chemical biology sides. And there are also other like 3D proximity-based DL, like Hypergen APS, which is in Denmark. They use these methods, and like there are DNA-guided methods, which was like earlier methods. Now like it's almost discarded. And encoded self-assembly libraries, it was an earlier method, but now it is like again it's coming up. So, but I'm not going to discuss all these methods. So basically, I'll discuss this DNA recorded method, but still DNA recorded method not in the solution phase, because this. So now we do modified this method into like solid phase. So I'll discuss this method and like it's how we have synthesized. It's in not, you cannot say like exactly solid phase. We termed is like pseudo solid phase, because uh, I'll tell like how we do in pseudo solid phase. Yeah. So we start from a live like DNA. This is the DNA, and that as 5 prime has like amino functional, and these DNA are on bead and like CPG protected. And so basically you have a 96 well plates, and each plates have these DNA like these beads. And now so you do parallel reaction, like so first phase we are attached like the amino acid like FMOC, DAP and MTT protected. So this here basically carboxylic acid and amine reaction. Now I have this DNA like FMOC, DAP, MTT, FMOC on, and like when we use like 3% TCA by DCM, like this MTT will be removed, so it will generate a free amino group, like free amino functional. So this point we are doing all in parallel. So now we are doing another set of reaction, basically attaching with this amine 150 like carboxylic acids. So and like when we purchase those like DNA amino functional, so this is the coding region. These DNA codes are fixed here, like, and this code represents this acid, and this code represents this acid. This way, like we have 158 different sequences. So we did this reaction, and then after that, first is we'll purify by HPLC and check their MS. Now, when if MS is correct, then only we'll pull it. So after that, we pulled it. So basically, you have 158 reaction parallelly. Again, you will pull it. Now, then you split this entire pool into another like set of plates. So how many number of reaction you want to do? So basically, in next stage, we wanted to do like 369 reactions. So we plate into like 369, split in different wells. So now why we're saying like it's a pseudo solid phase, because this after elution this DNA up to these are in solution phase. So now what we did, we used anion exchange resin. It's like DAE safer, because DNA is like negatively charged, so they will bind to this resin. And so now basically now this DNA is attached into this solid support resin, and you can do lot of solid phase chemistry. So now we did this same like solid phase reaction, like solid phase chemistry, like 369 acids. So after that, again, you can elute this like DNAs attached to its small molecule from these beads by doing a simple acid wash, like pH not more than like 7.4. And then, so once you eluted, now you have two building blocks, two sets. One is here, another is here. So you have encoded one set, like the first set, and the second set is still you need to encode. So then you will do a like PCR reaction, simple tagging. Like we did clean new polymerase extension to tag our like second set of like these building blocks, these here. So around 369 tags. And then you test your like different tagging reaction, how they have behaved, whether proper DNA length is there or not, and based on that, so you choose, and then you pull it, and so now this is your library. And even we did not purify it, but still here you can purify is a in a pool in HPLC. So for different library based on the chemical reaction yield and also this polymer like PCR reaction, sometimes you need to purify. For this library, we did not like purify. So like this is the encoding scheme in like detail. So basically this is the code one, and after doing the first and second reaction, now this is your annealing region will put, and then this is for second reaction will put code two, and this kind of clean new extension reaction I will do, and now you'll have the full double stranded DNA. So basically you started from a single stranded DNA, and now it's from double stranded DNA. Okay, so this is the one method like pseudo solid phase that we have worked. And now this is the another method. So still now like for major companies they use this method. This is entirely in solution phase. So basically what you do, you start from this linker. So it's called head piece, like bifunctional, and this linker, so it is full, then you split in like different wells, and then you do attach your another set of DNA. So by doing like tagging reaction, PCR or different kind of small biology, then you do simple another kind of chemistry like amino acid attaching, and then you will pull it. After pull it, again you split it and then encode it, second set, and then you do another reaction. So in this way you can grow, like you can go up to 3 cycle, but 4 cycle library is still there, but with 4 cycle you generate a lot of impurities. So up to 3 cycle library is best, and you can go up to like billions of or molecules, but trillions of molecules are also reported with 4 5 cycles. So now, like, how do you test your like kind of this QC process or how they are going? So basically, when you are doing all sorts of this PCR tagging and this encoding scheme, so each step, like you will run all those reactions in jails, and you'll see the band shift, how the band shift has been done, and based on this band shift, you decide the yield of your tagging reaction, and they'll go for it. And another thing is that before synthesizing the library, you will test each and every condition. So like with your fragments, with your DNA tags, so that when you are synthesizing your library, you will have a like very good yield. And this is like general encoding scheme with G4 DNA ligase. Sometimes we use a lot of schemes are there for like basically the conventional molecular biology. All these PCR techniques could be employed here. So that's like so it's a marriage between molecular biology and synthetic chemistry. So now I'll go to the QC. Like when you are synthesizing, as I already told that after first cycle we are not doing purifying. So basically after first cycle it's all in pool. So this is like you can purify your library in pool, like first pool, second pool, third pool in HPLC. And another thing, after synthesizing, like you can run in jail and you can see the how their band shift is going, because you are tagging with DNA and their DNA like base pair is increasing, so there will be band shift. And another thing is that you will check this library mass, like their average molecular mass, and by that you will go whether your library synthesis is successful or not. And sometimes if you want a very stringent QC, you can do also like in mass analysis of individual wells, like individual pools from in before doing second reaction or third reaction. That can you also do, and you calculate the average molecular mass. And then so this is like QC, and another QC is like which is very important. So after synthesizing this library, you will check my high throughput sequencing, whether each DNA tag is present there and how they are like distribution. If you have like homogeneous distribution, that means you have synthesis libraries well. So all tag like concentration, their number of copies are like almost same, and that is very good for starting selection. So when you're starting selection, if you have a like inhomogeneity in this DNA sequence numbers, so basically that is a bias toward your like sequencing or like selection results. So that's not good. So this homogeneous sequence counts for each library member, which is like very important. Now I'll go what are the like selection techniques or like screening techniques. So like a lot of screening techniques are available still now. In literature it has been published, but like most important or like mostly used is like immobilized protein in solution, like or you can like do incubate your library with protein and then immobilize that protein, like vice versa, which I already discussed like in my introductory part. So that's method. And there are also like others methods are like cell-based selection, in solution, activity-based selection, these micro like coupling with microfluidics. So these papers are there, but in industry we use this method because mostly this is robust methods, and these fancy methods, like one of the major problem is the reproducibility, and this you cannot scale up this method. So still now this method is pretty robust and scale up, so we use this method. And we start screening generally from like 10 to the power 5 to 10 to the power 8 copies of each DNA molecules that should be present when we are like start screening our library. And like basically after selection you compare the like perform the DNA sequencing fingerprints of a library like before and after selection. Basically you take a like only beads and then bead plus protein, and you compare their sequencing enrichment. So now I'll go into the details of that how we do selection. So in your like lab generally you can do like manual selection, but here we prefer doing like all automated way. So here we use the like Kingfisher magnetic bead particle processor, and we use like plates, 96 well plates, to make it high throughput. Like here each, yeah, this column, this is like each sample, like each proteins, and these rows are like different steps. So here this row we have used like kind of different like beads are there, and here like the proteins are there, number of different proteins. Suppose here this plate like 12, so we can use 12 different proteins or one protein with different condition, like different like their cofactor, their substrate, and this. And then this, so basically this pro beads will transfer here, that will incubate, and then it give 3 was here basically. And after this is the library incubation. Here automatically it will incubate library 1 hour with immobilized protein, because it's a magnetic particle processor, so it will process these magnetic beads from here to here this way. So after incubation we'll have again 5 was, based on that standardization, how many was you want to do, like 5, 3, 6, so was. And then finally we elute this library with heat. So basically those small molecules will be like released from proteins, and then we like take that solution, and then we use that solution for our downstream process, because that solution has DNA. Okay, so now this is the like DNA sequence that we get after like DNA like library selection. So and this sequence is only for like that library I showed, and with like different library this sequence will be different. So with this sequence like DNA will basically divide into two parts. One is like Q for qPCR readout, another is you do your PCR and this next generation sequencing. So you do like qPCR to optimize selection condition and like pre-evaluate selection efficiency. Why? Because this next generation sequencing, it's costly. So one batch of sequencing it cost around like 5,000 US dollar. So that's why you need qPCR to evaluate, to check whether your selection process is successful, or you need to optimize that process. So basically you do a serial dilution of your library starting point, and you do in the cycle, and you want to see like with how many cycles this gets amplified DNA, and make a standard curves like this kind of things. And this is the most important figure here, you can look at that. So basically this red one, this is like only beads, that this is after selection in qPCR, and like this black one is that target protein that is immobilized on bead, and then the selection result that we have amplified in qPCR. You can see that like after qPCR selection, so like around 19 cycle, after 19 cycle only beads, it gets starts increasing like accumulates. But here, like when library with protein, so you can see after 10 cycle it gets like huge accumulation. So it clearly tells there is like DNA enrichment difference in DNA with like in presence of protein. So and it tell us that okay, my like selection experiment was like successful. So with that things, and also like if sometimes you do lot of others experiment with reference DNA, small molecule DNA conjugates, which I'm not going into these details, so that will confuse a lot. So this tells okay, our selection is successful. So now we know our selection is successful. So we'll go into the next step. So next step is do what we'll do like 2 rounds of PCR. So I already completed the qPCR. So in first set, we'll attach 2 tags kind of here with near here. So because why? As I already told this, sequencing like one channel with Illumina TrueSeq or HiSeq, we can maximum read 150 million sequences, and that is also costly. So we can do like multiple experiments. So as I said in 96 like that plates, we are doing 12 experiments parallelly. So we can use 12 protein. So we'll be tag those one with our different codes, and then after that PCR will purify those each reaction, and then from those each reaction those DNA will be used as a templates, and we'll put equal amount for like second PCR, and these all things done in parallel. I'll do at a second round of PCR, and there will be attached another code here, like so basically those are Illumina prime, and they will have the final length DNA around 2002 base pair, and you will do purify all those DNA, and you will pull equal amount from like each reactions, and then you purify agarose gel purification, ethanol precipitation, like simple molecular biology conventional work. Then you will put out around 700 nanogram of DNA for next generation sequencing. That amount is good for like one lane sequencing with like one chip and 150 million reads. So now, so then basically your wait timing, so you'll wait for around 2 weeks, because sometimes if you do not have your own sequencer, that takes the library, their preparation and running. So after 2 weeks you get the result. So now it's the analysis of this result. So you get your result in like core server. So you have to core server to download this DNA faster data. It will be in genome Java. So then yeah, you can, for generally for academia they use like open source, you can upload into Galaxy web server. So Galaxy web server, lot of like open program is there, like Bash, Linux, R, Python program is there. You can use, you can comment, like you can get your data in fast queue. Then you will use Illumina adapter trim program, that is also available in Galaxy, and you have this data trim fast queue file. So now once you have this file, but here you, if you are in academia industry, that does not matter, you have to write your own code. So basically you can write your code whatever like language you want to use. We used like Python. So based on our like building blocks, they are structures like and there's DNA sequences, so you'll put those sequences as input, and we run that code, so that will analyze this data. And here like now here what we do, we have our own platform like Linux or Mac based like supercomputer. So we'll use like Bash shell script, like Bash scripts, adapter trim stats. So that will directly from our code server to, they will give this data trim fast queue. So you do not have to do all these sorts of open source stuff. This data will also protect it. So from there we have also here developed our own code, so that will analyze this data, and that will give our text format data. So, and that text format, so basically it will give that each like compounds, their structure and their now DNA, that their count, how many DNA numbers are present there, enrichment. And then you play that output with different cheminformatics softwares. So you can use Data Warrior or whatever, like Parkin, Alma, Spotfire or other platform, and then you get a sequencing finger plot. So how this fingerprint plot looks like, so as I already told, it's like it looks like a 3-dimensional plot, or you can plot in a 2-dimensional. So this is the like sequencing fingerprint plot against part 1. And I say like this is x-axis. This is represent the fragment one. It's like 158 fragments are there, and this is like y-axis. It's represent like fragment two with 369 fragments are there. So and like each sphere here like represents like one molecules present in our library, and the size of the sphere is higher, means their enrichment is higher. And this g-axis, this is the enrichment score. It's normalized sequence count. So basically our by color we have also represented here, because it was easier. It was a two building block library, so with three building block it will be little bit different. So this red color means which have maximum enrichment. So now if we look at, so now this entire cubes, it will be full of molecules. If you look at this data which is non-selected, so all this like around 60,000 molecules will be there, but here selection has been done. So you can see that this is a kind of one straight line, kind of one band has enriched upper, like one plane. So that it tells that okay, only our selection has been done around this plane. And that plane, if you look at this A65, this is the coordinate. So that means this is one of the important fragment which is playing some role in binding here. Now in combination with A65, like with other, what are the other parts, they are also like playing role. So we can look at the B101, this fragment with enrichment is higher, maximum. So based on that you decide, okay, so you have a lot of hits here. So with screen you can see that like, and these compounds are diverse also. So and the main compound is A65B101, along with like others. You can still consider these molecules are also enriched, because this is a kind of super good selection picture. So now okay, these are the hit, but still there this hit's not been translated from like on DNA hit to off DNA hit or real hit. It could be there is chance artifact. So you have to validate in orthogonal assays. So sometimes we validate in two assays parallelly or three assays, but here I will discuss only one biochemical assays. And you have to synthesize those molecules in off DNA. So basically, I have for the same chemistry, I have synthesized this linker in two step: attach POC, then removal. Once you have the linker, you'll do like attach a fast building block and purified HPLC, you check the NMR, and then again attach to your second building block, purify, check HPLC, NMR, confirm okay, our off DNA reaction is right. And so whatever the scheme you are following for your on DNA chemistry, you will follow for off DNA synthesis. That is very important, because if some of the impurities are being generated during that reaction, you can also tackle that. Okay, this some in off DNA reaction I am getting a major product which is not our desired product. So that will help you to understand your downstream like results, why you want that heat is not being translated, this kind of questions. So now we'll go to the off DNA like heat validation. Okay, so this is our heat validation against part one. So if this is the two-dimensional plot as I have discussed. So this is y-axis is fragment one, and like x-axis is fragment two. As I said already, our this we got a one straight line, and this coordinate is A65. This A65 is the main structure, along with this is the main heat, and these others. So you can see the lot of structures we have enriched, and these were structurally diverse, and some of the structures are novel. But this structure A5, like 153 and B356, these are not novel, because this structure, this fragment was already reported in like some of the existing PARP1 inhibitor. And other than other structures are like really novel, I can see that IC50 are like pretty low, 108 nanomolar, 190 nanomolar, and this is simply direct heat from your library, and even before doing any medicinal chemistry optimization. Then you also got some new kind of like novel chemical space here, these kind of compounds. So now with that aim, so as I already in my introduction part I told that our aim to generate like one single library, and we'll screen multiple proteins in the same class and see whether they can deliver the targets. So to test our like hypothesis whether it is successful, it is time. So what we decided, we decided to screen the other NAD binding protein. So basically we screen other ADP ribosyl transferases, kind of PARP 10, 12, 14, 15. We also screen like PAR 4 and PAR 16. So unfortunately, we didn't get any hit against part 4 and part 16, but we end up hit against these 4 parts. So these are the all screening fingerprints against there. And as I already mentioned, these are all 3-dimensional plot, like x-axis and y-axis, and each sphere represents one compound. And this g-axis is the enrichment score that is higher, or red color is that is the maximum enrichment compound, and their affinity will be higher to the protein target. So in this case, you can see clearly 3 enrichment compound against like 10, part 10, or you can say these 2 you can also consider. But low enrichment, sometimes we do not consider as we have superior compound. For part 12, there's this one compound we focused on. So we could have focused this one also, but we consider for just to test our hypothesis this one. And part 14 also we got nice fingerprint. So you can see that one band is there. And for part 15, we got like lot of molecule, you can see. But here like this, mostly this was the maximum a enriched molecule, and you can say each case you can there 101, this one coordinate is there. So and so you can get lot of structure activity relationship data from this one, which I'll discuss in my like coming slides. Okay, so this is the screening fingerprints of like part 10. So these are 2-dimensional plots. This 2D plot you can see like it is in, this is fragment 2 that is in x-axis, and fragment like 1 it is in y-axis. So now you can see this straight line, so this part 10, so like B354, so this has been generated. So from here we can see, okay, that means this fragment is important and it's playing a role. And the maximum enriched compound is with this one, red one, A34, and the second one is our A82, these others. And another thing is that you can see also like one line has been generated here, which is A47. Because why we looked at, because this fragment, this was earlier already reported against parts 10 screen. You can see, okay, this fragment is still showing up some molecules. Now, if we have any other better fragment pairs or modification which could turn into a better molecule, we could say much more enrichment after covering these new, like these new molecules. So here, so these molecules are structured in novel. So you can see like tetralone containing moiety, and we have like validated those heats in biochemical assay. These are biochemical activity 6 molar micromolar we got for this one, and that second enriched it was around 25 micromolar. And like when you docked it, you can see this one, it is targeting not in active site, it is like site new pocket. So like it opens a like new dimension. So basically you can develop these molecules from this tetralone and site targeting pocket. So now we'll discuss that other protein. This is like part 12, as we already mentioned. Like you can see this, we are getting 2 bands, but still here their enrichment was not high, so we didn't focus on these 2 bands, but instead of focused on these compounds which have like high enrichment, and we synthesize these molecules of DNA. And another thing is with DNA we have the linkers here, and sometimes these linkers have a role in like affinity. So you have to like tweak it little bit, or like moderate this linker and see like how they are behaving. Here you can see with propyl also we are like this activity around 33 or 34 micromolar, but with carboxamide this activity is like around 100 micromolar. And another things like we can see that most structure activity relationship pattern is all paracarboxamide. This kind of like SAR is giving this, it is like carboxamide, like but it is in para position. So now we'll get like par 15. So par 15, like I would say this is the still now uh, the molecule reported against par 15, which is the lowest IC50, and this heat is directly from encoded library without any medicinal chemistry optimization. So there is a huge scope for developing this molecule from the starting point, because it's already quite low, 200 nanomolar, just delivering a starting point. So now before going to, I'll discuss this fingerprint. So it's a 2-dimensional, as I already mentioned, because, and another thing that this 2-dimensional plot is possible here because it's a 2 building block library, as I already discussed. So this is fragment 2 and this is fragment 1. They are coordinate. You can clearly see like two band is there. One is like on B114, this here, and another is A101. So it clearly tells, okay, these two fragments are more important and their combination has reached enriched maximum. So check there, huh? So this is around like 970 nanomolar. Like this one, like second best compound, it is exhibiting like 200 nanomolar with toluidine compound, and this A101 thiolhydrazide fragment. And also when you look at this their docking study, you can still see that, okay, this is targeting the catalytic side because carboxamide, and it is that part, this part is occupying like some sub pockets which is like away from catalytic side. So basically we're targeting both the pocket. And so this is like, as I already mentioned, like first reported part 15 selective inhibitor directly from encoded library. And another thing, part 14, this is the results. Same thing we got, like you can see a band. So it's clearly okay. So this fragment, this is important, like B259. And we didn't want to validate this molecule because when we are synthesizing this library, that time there's as a report of this molecule against like par 14, so which has this fragment. So and basically that also validate us, okay, our whatever hit we got, it is real because it's already reported. And so we did not further went for validation because that molecule that time was that they already published when we were like doing the selection process and all stuff. And it also the same thing, kind of meta substrate carboxamide, that SAR has been delivered. Okay, so now another thing I'll discuss. So I talked a lot about this chemistry, how do we screen and these things. And another most important thing is that for doing screen, you need a protein, and that protein should be good quality, kind of functional and solubilized protein. And how do we express that protein? Because you need the protein which is biotinylate tag. You can do the screen with other tag also, but with biotinol tag, so you get the most good result. So that's why we prefer, not only we, that in this field maximum industry and academics, they prefer biotin tag protein. So to express this biotin tag protein, you put your like insert, like here we have expressed SAT26 in-house SAT6 protein, like in your vector, and you put the avidin tag in N terminus, and you put S like in C terminus, you'll automatically get his tag from this plasmid. So after expressing, you will purify through HPLC like his tag, and after having this like his tag, you will immobilize against your biotin tag. Another thing, like you co-transfect this plasmid with PV resicum, like expressing plasmids in cells, and you will supplement those bacterial cells with biotin, and they will like conjugate biotin into this avidin site. So after purification, you have fully biotinylated protein. Once you have that biotinylated protein, you will do characterization of this biotinylated protein by LCMS or like colorimetric quantitation, how many biotin are there. So when you do in vivo biotinylation, mostly you will get specific one biotinylation, and then you will check the like whether this protein is still active in after attaching to bead. So suppose here for sartwin, this is the kind of one kind of assay. So you take a one peptide which is acetylated peptide and put sartwin enzyme, it will deaccelerate that peptide, it will form this so now if you run in HPLC, those two peptides, they have different like elution time because their polarity is different. So basically this is the H3K9 deacetylated peptide, and this is the myristoylated peptide, this is the control. And with protein you can see like both peaks are getting, so that means protein is active. So now to confirm that, what you basically will take your biotin tag protein immobilized on bead, and then we'll treat that immobilized like protein on bead with these peptides in assay. After that you will like run the supernatant from that assay in HPLC and check, okay, you are getting both the two peaks, that will say, okay, your protein are active on bead. So now it's a green signal, good to go for your like Dell screen. So we'll put those protein for Dell screen. So now we have active protein on bead. So we'll go for a Dell screen. So basically it's a like further application of this technology, that how we can use this technology for like to discover inhibitor. Not only inhibitor, you can get activator or different kind of sort to somehow mode of action. Like if you look at this mechanism, this is this active. So like twin mechanism, that how they deacetylates. So if you take a like thiacetylated peptide instead of acylated peptide, so they forms this alkyl mediate, this complex, and this intermediate instead of like oxygen, if you have a like thiol here, so this is stable. This intermediate will be stable, but with oxygen it will be like hydrolyzed and it will get very quickly and you will get a deacetylated peptide. But with sulfur thiacetylated, it is stable. So now we wanted to like discover small molecules against SAR twins with different function, because if you look at the SAR twins, they have like the acyl channel, and this is this NAD binding pocket, and this is like basically substrate binding pocket. It's called acyl channel. So for myristoyl, it occupies here, and for acetyl, still there is like space is left here. So now we want to discover some molecules, so basically which will specifically inhibit the myristoylation but not this deacetylation, because deacetylation they have role in DNA damage and others response in cancer, and this myristoylation they have role in longevity and aging aspects. So basically we wanted to molecule which specifically inhibit this deacetylation, not demyristoylation, or sometimes demyristoylation but not deacetylation, or this will activate the function of this like demyristoylation. Now to do that, what our plan was to like screen our this library, this protein, and this protein plus like this NAD, like non-hydrolyzable NAD, and they are like different kind of complex acetylated thiacetylated peptide, thiamyristoylated peptides and their combination. So kind of different shape will be created in that stage, and then fish out that chemical shape. If any molecules in your library fits, so it will give that molecules, and from that hit you can validate in functional assay. So now why I wanted to so this is like one kind of advantage of this technology. So you can't do this approach in conventional medchem. Basically in conventional medchem, from screening then you have to optimize that molecule in particular assay, interrogate and develop. So you can impose like sort of mode of action study on early stage of your discovery. But here, like here, if we want to target develop any molecule against aging, so basically we'll target this mechanism of action, like we'll first develop this assay and this mechanism, and then we'll screen in that condition. So now we are here, okay, if you are getting any molecule, it is going to work in that way. I'll get effect in like longevity pathway. But this kind of research doing conventional medchem, it takes a lot of time and a lot of money and lot kind of basically resources is very high. But with this following Dale approach, you can do with less time and with less resources, kind of with less investment. So now this is our screening fingerprint. We can see that one molecule we got and we synthesized off DNA, and this was the first SAT6 reported small molecule, and which in terms of like less than 10 micromolar activity. And we have that crystal structure of this molecule, and we got this molecule like in presence of myristoylated peptide but not in acetylated peptides or other condition. And like this crystal structure is unpublished, and it clearly tells that okay, whatever the condition we have screened exactly fulfill that way, and that binding is not conventional into like acetylated peptide binding pocket. It binding is totally unconventional, and this part is like they're targeting that pocket, basically which known if you target that part of that protein, this protein will get activate. So now like in this program, like in my previous lab, now they are doing medchem modification from computational with this structure, so that it can bind this pocket much more prominently and it could turn into a activator. So, and now this is the all sort of screening that I'll go through, I have discussed. Now I'll go into the big question which comes to selectivity. As I already mentioned, okay, our library is like focused library. So basically, if it is a focus library, so there's a high chance we'll end up with less selective molecules. So now to do that, what we did, we just merged all our fingerprints, like screening fingerprints for against PARP 1, 14, 12, 16, say, and this is a 2D plots. So now we can see that the screening fingerprints are quite divergent. Suppose if we look at this PARP 14, this is the PARP 14, and for PARP 15, you can see this is the main bands here and like these two bands, and for part one here. So and this so and when you do the selection, even before doing off DNA validation that I shown earlier, actually we did this kind of screening fingerprint merge. So that will give you confidence, okay, whether I should go for off DNA synthesis and further validation, because I know, okay, these are the hits, they are going to be my target selective, these are the hit, these are going to be off target selective. So based on that you can choose, okay, this whether you are going to work on or not, and that kind of luxury you do not have in your conventional medchem. So now this is like profile our selectivity for SART. I'll discuss the SART6. SART6 you can see it's highly selective, and like we use 10 micromolar. SART2 3 other as very less, and for this PARP 10 also same, this PARP 10 others are very less. And for like PARP 1 we got heat also against like PARP 2 and like PARP 11, because they were the PARP 10 a PARP 11 2 and others were basically their structure, protein structure is highly similar, so it was not that much selective. But in case of PAR 15, you can see that that is the best molecule. We have 200 nanomolar activity directly from library. It is highly selective. We only got hit against PAR 10 because that catalytic site has somehow quite very similar with PAR 15 and PAR 10, but still we got a good selectivity, and little bit of medchem, applying medchem, playing around, it can turn into very selective molecule against like PAR 15. So now basically I'll like conclude here. So this part and this part. So basically we have designed a NAD binding protein targeting library, and we called it NADL. And so it's a target class specific approach, and we have successfully demonstrated that with low cost you can generate a library like within the academia, like academic sort of investment, and that can still deliver you a good screening campaign result and reproducibility. And from these results, like we got the most potent inhibitors against like PAR 15 and SART6. So now I'll show you much detail about the like experimentation that I do in like couple of slides, and then I'll finish my talk. So if you look at this here, so as I already mentioned that some kind of you need tag, we prefer biotin tag. So this is like biotin tag screening. You can see this much more like screening, stringent screening, but here this is his tag. You can see a lot of molecules. So this is noise out there. So and then is that before going into the screening condition that I said, like we do use magnetic particle processor and those conditions. So you do a lot of optimization experiments, like in what condition you are going to use. And another example I will give you. So basically here protein concentration, that concentration you are using, that is also very important. If you use very high amount of protein in your screening, so you will end up with the heat which is high micromolar. You are not going to get this nanomolar heat, and that in if you're using a low like nanomolar amount of protein, you will see those inhibitors which are like high kind of low nanomolar activity, so they will enrich kind of high their score. Suppose if you look at this seconds plot, so here this is the screen using 1 micromolar PTK, 1 micromolar BTK, and the biochemical activity is 2.5 micromolar, 2.5 for one this molecule, and you can see the enrichment is higher in 1 micromolar, but when they have is 50 nanomolar, so like this molecule is like 3, it has like around 500 nanomolar, you can see their enrichment is higher. And another thing is that, so when you're doing this screen, you can use like the ATP kind of, what kind of cofactor, what kind of substrate there's kind of that mechanism has. And from there you can already come up with inhibitor with mode of action, like okay, okay, this is going to bind this kind of thing. So suppose if you look at this screening pattern, so for this is the only protein this has been enriched, okay, higher. But in presence of ATP, this one, this has not been enriched. So that means this compound is binding into ATP binding site, but not here. And these two compounds, they are binding to other site. They are not like competitive binding with ATP. And suppose when you are using known compound, so using that no compounds are in this. So that means they are like sort of action is quite similar with the known compound. So, and this has also been validated this for like third compound, three structures. You can, they're from their crystal structure, you can see it clearly, like it does not bind to this ATP binding pocket. It extends its binding to like other pockets. So now I'll like finish my sessions here. So like there are multiple parameters like kind of type of protein tag, solid support, was that influence your enrichment and compound. And another thing is that your protein concentration is very important for your selection, that depend on the target affinity. And there is a selection results, like so from your selection result you can get a good amount of SAR even before starting your med chem you can have. And that is the one plus point, like one advantage of this technology. Another thing is main point is this technology is it takes very less time compared to high throughput screening, and with very like less amount of, it requires very less amount of investment. So basically if you want to screen a 1 million compounds library in corporate library in high throughput screening, so it will roughly cost around like 2 billion, 400 million to 2 billion. So based on your like number of compounds, whether that they are available or corporate. If it is entirely corporate library, it will cost around like 2 billion. But like for 800 million DNA encoded library, within 150,000 you can synthesize this library and screen and all sort of, you can come up with it. So basically cost like around 400 to like you can see point less than $1. So that is the most two like advantage points of this technology. It's like it takes less time and it takes less resources. So I think that's all for my talk, and I'll finish up here. I'll quickly go through two slides. So basically, this type of steel targets have been like integrated by Dell. So you can see a lot of targets. You can like kind of type of targets you can do by employing Dell. And then I'll give the success stories in Dell, because this technology is here for more than 10 years that has been established, and thus first it was pioneer in 1920s. So these two example, this is from XChem. So this is like ENPP2 inhibitor. This is also going to phase 1 clinical trial. It directly comes from Dell library. You can say the 250 to 25 million library. It's a 3 building block library, and you can see this is their direct hit molecules, and this is their like final the molecule that is going to into clinic. You can say that like this part has been off, that was with tag DNA, and like this part is still there, and little bit modification is here. You can say that medchem effect is very less. And for GSK, so from 7.7 billion they came up with these heat molecules, and this is the like molecule, this is going to clinic, and this was the heat structure. Basically it's simply one modification here, and they developed this molecule after screening to like up to clinical stage for phase 1 just within 6 months, because downstream medchem was like very less. Okay, so still now this is a summary of that main important papers in this field. So see the 1985 it was like fast display, and then they Lerner Barn the 1992 fast conceptualized. Now the field has like gone through it's a quite substantial amount of paper is there, and that's why like industry has started using this field intensively. Now it's a like robust and solid proven field that okay, it can generate a lot of good hits with low cost and investments. So And now this is my acknowledgement. So I acknowledge like my previous lab with this entire work I have present here. It was from my previous lab, Professor Rafael Franzini from University of Utah. And then I also work with Professor Sunil Sharma from TGen, and also obviously that my present affiliation, Orion Corporation here in Helsinki. I think that's all for the day.